Hi, I'm James Crow at Vanderbilt University, and I'm going to tell you today about work we've done with human monoclonal antibodies to avian H5N1 influenza. This work was done by scientists at Vanderbilt and great collaborators also at the CDC and at Scripps Research Institute in La Jolla, and also at the University of California, San Diego. So we put together an interdisciplinary group to understand how human antibodies kill avian influenza viruses. You might remember in the past year or so, very controversial papers were published about the passage of H5 influenza viruses in ferrets. Previously, it was known that bird flus like H5 pass from birds to humans, but they typically do not pass from human to human. And the question in the previous studies was, why not? And several groups in the Netherlands and Wisconsin and other have done, in other places, have done studies to passage those viruses in ferrets and to cause them to become transmissible in mammals. And by inference, these viruses might pass from person to person. These controversial sequences were published in Nature and Science, and we looked at those sequences to find the place on the influenza hemagglutinin protein on the surface of the virus to understand exactly what the changes were. Now, we've been studying experimental vaccines for bird flu, and we've looked in human beings at the B-cell response to those vaccinations, and we've isolated human monoclonal antibodies that kill conventional H5N1 viruses. We tried to understand, would these antibodies also kill the altered viruses, the ones that are mutated so that they're transmissible from ferret to ferret or human to human? And in fact, we found that these antibodies do kill the altered viruses. But the question was, how do they do that? Because many of the transmissible transmission mutations occur in the rim of the receptor binding domain, and we could tell from our tests that the antibody bound to the receptor binding domain or in that general region. It was not clear how mutations could occur but not abrogate the binding. So we used a number of different studies, including escape mutations, deuterium exchange mass spectrometry, electron microscopy, and crystallography to determine exactly where and how these antibodies bound and killed influenza virus despite the mutations around the receptor binding domain. And we found that some of these very potent antibodies have figured out how to slot in to the receptor binding domain without touching the rims of the canyon that surround the receptor binding domain. And the rim is really where the transmissibility mutations occur. And the antibody has figured out how to slot right in between those and still interact with influenza and kill it. So the studies elucidate the molecular basis for neutralization of these somewhat scary viruses that could occur naturally in birds or humans, and also suggests that conventional H5 vaccines, the subunit vaccines that are being studied currently, most likely would induce these very potent clones that know how to engage the hemagglutinin without being disrupted by these changes. So that's very encouraging, and also supports the rationale of publishing these types of studies about escape mutant viruses, because using that previously published information, we were able to determine molecular mechanisms of immunity, which is very helpful to vaccine design. Now, the techniques we used could be considered a hybrid method, because we were trying to understand the interaction of the antibody with the hemagglutinin protein, and so we used both imaging as well as molecular biology and proteomic studies to gain information about the epitope. And then in the end, we used Rosetta docking computational methods to fit the structures of the hemagglutinin and the antibody together in a way that we knew they should interact. It's really the computational modeling that brought it all together and explained the mechanism. So in conclusion, it's very encouraging to know that subunit vaccines for H5N1 should induce antibodies that protect against highly transmissible variant viruses. And also this work elucidates how you can put together a hybrid method approach to understanding epitope mapping for antibodies and complex structural epitopes. Thanks for listening.